pleased tonight. We are very pleased tonight to have a speaker tonight who's going to talk about the American Chestnut Foundation. So John Scrivani studied forestry at Clemson and Oregon State University. Oh, we have Beth Lumberger would like to read a statement first before I introduce the speaker.
Sorry. This is a personal statement that I wrote because the board hasn't made a permanent decision about any of this, but Bob and I both attended the DEI, is it DEI? And a Native American lady there asked us if we would begin our meetings with acknowledgement. So this is something I wrote this afternoon. This land where we live was historically Tutelo-Monacan. As a barefoot Appalachian child, I too felt the earth I walked on and was thankful for the wonder of nature around me. Now, as an older and sometimes wiser Appalachian, I'm learning a new way of managing the land for biodiversity, which connects me to all who live on this land and will live on this land. Whether your ancestors were Melungeon, European or Asian immigrants, enslaved Africans, Native Americans, or from anywhere around the world, this gift of native habitats that were managed so well for tens of thousands of years by the Tutelo-Monacan people now needs all of us working together to preserve it for future generations.
That's one of the best land acknowledgements I've ever heard. Thank you for writing that. This is Melody Mobley. I'm the chair of the DEIJ committee.
All right, so our speaker tonight, John Scrivani, studied forestry at Clemson and Oregon State University. After earning his PhD in 1985, he taught forestry at Virginia Tech before taking a job as a research forester at the Virginia Department of Forestry. John managed the research, tree improvement, forestry inventory, and information technology programs. John was first involved in American chestnut restoration with breeding work at Lasagne State Forest in Nelson County. I had to ask how to pronounce that. John continued that work while at forestry and after retirement as an active volunteer with the American Chestnut Foundation. John is currently the Virginia chapter president and also serves as restoration chair on the board of directors of the national organization. Let's give a big welcome to John.
Thank you, Bob. Let me get this set up. Thank you all for inviting me and talk about my favorite subject. And as Bob mentioned, I have a fair amount of history in it. So I'm going to go ahead and share my screen if I can. Okay. Okay, are the folks online seeing? Well, let's see.
It says you have started sharing your screen, but the slides aren't up.
Okay.
Oh,
Are they up now?
I can see like all of PowerPoint. It's not in presenter mode.
Okay, let me go. I thought I was. Oh, I need to share the other screen. I believe you might be seeing it now.
No, it's
No.
back to the black screen.
Oh,
Oh, now it came through. Now it's just the slides.
okay, a little delay. Okay, great. So yes, so as Bob mentioned, I'm Virginia chapter president of the American Chestnut Foundation and also on the national board chairing the restoration committee. So our organization is dedicated to, well, our mission is right up here, to return the iconic American chestnut to its native range. And our vision is a robust eastern forest restored to its splendor. both, I like to make the point that both of these, achieving our vision and mission require that we collaborate with a wide and diverse community of forest stewards. Including, as we had the earlier, the land acknowledgement from the indigenous communities. They're going to be key in this as well. And anybody who owns land, manages land in the Appalachians and in the broader Chestnut Range will be key for us to successfully get this very key foundation species back into our eastern forests, particularly in the oak chestnut regions of the Appalachians. So, let's see. Should be able to go to the next one, and I am not for some reason. Okay, it's my
You might have to click and then you can use the, yeah.
Yeah. So, American chestnut, Castanea dentata, very distinctive leaf. Once you start studying chestnuts, you can tell purely or wild type Americans pretty definitively from Chinese and Japanese and first generation hybrids. But when you start getting advanced generation hybrids, they get more difficult. But this is a classic leaf right here. It's a canoe shape, tapered at both ends. This one doesn't have that prominent of a leaf tip, but it's often has more. It has the teeth, hence dentata, and they are they have a little forward bend, a little breaking wave is the way you think about it. They are pretty much hairless underneath. The Chinese chestnuts have a lot of white, a thick mat of fine white hair underneath. There are some fine leaf hairs you can find with a hand lens on the midrib in the American. That's actually diagnostic. They're a monoecious species. They have male and female flowers, male and both in catkins. The males only catkins are the ones that shed the pollen first. The females, I don't have a good picture, maybe later. There's the female catkins are further behind. They don't start shedding pollen. There's female flowers at the base of those catkins that look like little pineapples, and they get pollinated and they turn into burrs, and they have typically 3 nuts in the burr. Sometimes you find 4, 5, 6, even 9, but typically it's 3 or 2 or 1, depending if some abort. Very distinctive tree. It was a tall tree, fast growing, also very well adapted to the eastern forest. So we don't really know the exact range, native range, which is actually kind of a fluid concept anyway. So you have to have a point of time. If you go back to the post-glaciation, at the peak of the Wisconsin glaciation, the range of American chestnut was the Gulf of Mexico and parts of the Atlantic shelf. And so after the glaciers receded, it started moving back up but fairly quickly got up into the Appalachians, particularly the Southern Blue Ridge, up even into this area. And then for a number of centuries, a few millennia actually, it was slower in moving up the Appalachians compared to like oaks and hickories up until Maine, but eventually did this. The dark on the left, the darker green is representing more or less what was considered to be the native range pre-blight, and then there was the lighter shade of green that extends further east and west and south, was more or less where it was probably within the last 500 years, possibly. But this map on the right is a pollen record. It shows how much chestnut pollen shows up in pollen cores from ponds and other ways they find out about forest composition in the past. And actually the colors represent 1% of the pollen, 1 to 2%, 2 to 4%. And that doesn't sound like much, but compared to chestnut doesn't shed a lot of pollen. And so they adjust these, they have to adjust these pollen counts to account for the different pollen production rates of different species. But anyway, this does show where they were most concentrated, as you can see, up the pretty much the whole length of the Appalachian mountains, also into the upper coastal plain in Maryland, up into New Jersey, up into Massachusetts, Connecticut, and then up into Maine. And if you go back the full 20,000 years of pollen records that are easily accessible, at least, it's been up into Canada, it's been further south, been down in Florida, it's been around, but this is pretty much representation of what it was before European colonization, at least. And in fact, looks like a lot of land clearing, particularly in New England, actually fostered the spread of chestnut into parts of, or extended the amount, the percent in New England. And so the dominance, the key foundational aspects of American chestnut was recognized by Lucy Braun, who in the 1940s, and she published this document, or this book, The Deciduous Forests of Eastern North America in 1950. Still editions being published every so often. It's a very interesting book. Describes in detail where a lot of the different types of forests were. And she recognized the oak chestnut region. She actually, the chestnut was so dominant in this region, it deserved to be co-dominant with oak. So it was the oak chestnut forest region. It includes most of the Ridge and Valley and Blue Ridge provinces, and into the Piedmont, and then onto the coastal plain from Northern Virginia northward and onto glaciated territory in New England. Chestnut, red oak, chestnut oak, and tulip tree are the most frequent dominants in this, as she described them. And she actually has, in this book, she has descriptions of plots she used to characterize the vegetation. And one of the key plots she used for the oak chestnut region, one of about 5 or 6, I think, was up at Mountain Lake. And that actually showed that plot. It shouldn't be taken as a representative of all of the forest here, but that plot had 85% chestnut, and had tulip tree and red oak were the other two, main other two that were in that. And they were big trees. And that's actually somebody at Mountain Lake, a scientist a number of years ago, relocated that plot, at least as best as they could, and found what's there now, which is mostly oak and tulip tree. So, which is typical. So one thing I just recently did was to map her, kind of overlay her map, which was not very detailed. It's really pretty good, but not at a real detailed scale as these current ecoregion maps that come from US EPA. And there's a really good overlap. I didn't include this slide, but if you threw her map over top of this one, it would correspond extremely well to these particular highlighted in blue ecoregions, which include the Blue Ridge Mountains, Cumberland Mountains, Northern Ridge and Valley, Allegheny Mountains, Northern Appalachian, Piedmont, Lower New England, and the Hudson Valley. And kind of jumping ahead to another slide, there are forest inventory data available now that we can use to get an estimate of where the existing population is. And within these ecoregions, which correspond to Lucy Braun's oak chestnut forest, 92% of the trees found in the FIA data are in those ecoregions. There are all these other, they're out there scattered, but the real concentration is in the Appalachian Mountains. And one reason I did this is we've been saying for a number of years, early foresters, when the chestnut blight came through, documented, or they reported that there were 4 billion trees that were lost. I think it's a pretty good guess, but I think that's all it really was. They didn't have solid data to say it was 4 billion. It was easily several billion, could have been more, depending on how you count trees. They also said 25% of the eastern forest, which was another guess, because in many places, like up in Mountain Lake, it was 85%. In other places, it wasn't found. There are some state level inventories that showed, 25, even higher in some areas, but it was very important and very concentrated in the Appalachians. So the current population estimate, and this is something I did running through the forest inventory data, which is collected by the Forest Service, and based on, there's one plot every roughly 5,000, excuse me, roughly 5,000 acres. And so there's a lot of potential to get a number of trees into the database. It's still very sparse. It's what's called functionally extinct, but there's still a lot of sprouts out there. You probably know if you're out in the woods, you find chestnut sprouts, and particularly in the Appalachians. So the estimate is, with some rough degree of accuracy of 30 million, is 431 million trees. If you say trees are at least a foot tall, so we would count little sprouts if they're at least a foot tall, and this is their protocol for all seedlings, and trees that were over 1 inch. So there's 360 million trees that are greater than a foot tall and less than 1 inch in diameter. So there's a lot of those sprouts out there, and there's roughly 71 million trees that are bigger than 1 inch in diameter. And when you start getting into higher diameter classes, the numbers obviously keep going down and down and down exponentially, but you still end up with estimates of trees up to 20, 30 inches of thousands, and in the 5 to 10 inches, tens of thousands across the range. And the colors here represent relative density in terms of stems per square kilometer. And there's areas where, this surprised me, but I've seen these areas where you have 4,000 to 7,000 sprouts per square kilometer. And if you hike along Appalachian Trail, there's places where you could not count the number of sprouts you see. And then there's also other records, herbarium records, that are showing up in this map, showing that trees were in Michigan. They probably weren't in Michigan very much when the blight came through, but a lot of people have been planting them, and they spread on their own, spread in Wisconsin, and a lot of people have been planting them. I mean, people plant them on the West Coast too, and a long time ago. So there's a lot of kind of human migration probably went on during period of European colonization. So, in pre-blight. So, not many trees, we call it functionally extinct because not many trees get large enough to flower, and the ones that do, the tree does not self-pollinate, so you need other pollen fairly near. Generally, if there's not something within a few hundred feet and it's flowering and you find burrs, the burrs very, very seldom have sound nuts in them. They haven't been pollinated. In Virginia, the statistics, Virginia, about 79 million seedlings and trees left, about 364,000 that are 3-inch diameter and larger. And it's not unusual to find a 3 or 4 or 5-inch tree. And they're fast growing trees, and every so often there are large survivors out there. And we have a database of large survivor trees across the whole range. All the chapters participate in this, and we have several thousand of those. And in Virginia, there are, I think, about 450 or the last count. And I've been tracking these things for 30 some years, and I keep finding new ones, and I keep finding some of the old ones that have died. So they are just fast growing. If they escape the blight long enough, they can get pretty large, but the blight may get them anyway. Now we find some trees though that I know of that have been around for 30 years since they were found to be large trees, and some of them seem to have blight resistance, and we can kind of tell. Actually, if I find a large surviving tree that has no sign of blight whatsoever, I'm kind of disappointed, because that's one that I'm probably going to come back to in 2 or 3 years and it'll be dead. The blight finally got it. But if it has blight cankers and it shows some level of resistance, those are, at least some of those can stick around for a good while. And we have some trees, some large trees that have been known for 30, 40, 50 years. So, yeah, we make a distinction between large survivor Americans, which are large, but they also have been surviving the blight, meaning they had been challenged by the blight and they're still alive, versus escapes, which are trees that are just lucky and fast growing and got big before the blight got them. So what does the Virginia chapter do? So, and I'll get more into the whole breeding strategy that we've been working on for 40 some years in the Chestnut Foundation. But the chapters help. I mean, one simple way of what the chapters do is we help preserve the germplasm, as we say. We help preserve the genetics by finding our survivor trees, putting them in orchards to either by getting nuts or grafting to keep them alive. We breed them with back cross program with Asian species to help keep them alive. And we allow them to sexually reproduce. So both with, in some cases freely, but also by controlled pollinations where we're trying to get better genes into those trees to give them more blight resistance. And this is actually where I first got involved in chestnut. This is Lasagne State Forest in Nelson County. It's just downslope from Reed's Gap, Wintergreen. If anybody's been to Wintergreen Resort, it's actually halfway in between Wintergreen and Devil's Backbone Brewery. So we've had a few after event meetings there at Devil's Backbone, but so this was actually donated to the Virginia Department of Forestry in 1968. And for the express purpose, it's about 419 acres for the express purpose of chestnut research. The Volks from Maryland did that, and this is a planting up here that dates to 1968. Actually, this orchard up here, and I don't know how well you can see it, but there's like white or creamy flowers. I took this picture in the middle of pollination season, because that's why there's bags on some of these flowers, and they are blooming. And this was something that people described about the mountains when chestnuts were here, was that it was like snow on the mountains in June and July, because if you had a solid chestnut stand, it looked, yes.
Yeah.
It's ammonia-like, it's not very pleasant. It helps you find chestnut trees in the wild. So, these are high. This was the Connecticut Ag Experiment Station started with hybrids. They were actually looking. I'll make a distinction between backcross hybrids and hybrids, but they were looking for just hybrids that could be 50% Chinese and 50% American, and they started with that, and then selected those to have blight resistance and growth. And they had some good selections. So we brought down, I wasn't around then, but the Department of Forestry and Connecticut Ag Experiment Station brought down 10,000 of these selections from the Connecticut Ag Experiment Station and planted them on this 10 acres here. And now we have hybrids. And over the years, my predecessor and I selected the best looking trees in both terms of blight resistance and growth. And we have some really nice trees that are 70 feet tall, 18, 20, 24 inches. And they are, not all were blight resistant, but some of them are pretty blight resistant. But one thing, and I don't know if you can see, there's a few little brighter green browns popping up. Those are tulip poplar. And we tried to control those for 30 some years, and they still get there and outgrow the chestnuts and the hybrid chestnuts. And one thing that we know from the old silvicultural text about chestnuts was that on more xeric sites, the poorer sites, on most of the between xeric and mesic sites, chestnut could outgrow tulip poplar. Only on the really cove hardwood sites where it's just too moist and too fertile for chestnut, that tulip poplar would outgrow them. So that was one of our indications that the hybrids don't have the growth characteristics of American. They can get blight resistant enough, but they won't compete in the Appalachian forest with, certainly not with tulip poplar, maybe not with northern red either. We didn't really test that. So the idea was, and Charles Burnham, who is a corn a geneticist, he used backcrossing to transfer genes from a resistant species to a non-resistant species for corn rust of something, or some kind of fungal disease of corn. So he came up with the idea that you could start with a 50-50 hybrid and then backcross it to an American. And so that generation would be on average 3/4 American. Backcross it again, you get 7/8. Backcross it another time, you have, third backcross, you have 15/16, which is kind of, in a lot of breeding, that's kind of the standard for true bred. You don't have to be 100% of any species to be considered true bred. In a lot of breeding programs, 15/16 is considered true enough. And the thought, and there was some early indications that there were only 2 or 3 genes involved in resistance. And the backcross breeding program was designed to, after 3 backcrosses, they did enough crosses and planted enough trees that they should be able to select for that very small percent that would carry both copies of all 2 or 3 genes. And so this was actually, these trees in the foreground were actually, I think we're looking right here at a 3rd backcross, and there's 2nd backcrosses, and further to the right are 1st backcrosses. So the Virginia Department of Forestry started backcrossing about the same time that the American Chestnut Foundation was founded to implement, among other things, to implement the backcross program. And the idea of the chapters is that each chapter throughout the range would do the same kind of backcrossing scheme. And so when we're done with the 3rd backcrosses, we would have a diversity across the whole range. We'd have varieties adapted to every region. And unfortunately, we found out, well, yeah, I'll finish that story here. Unfortunately, we found out there were more than 2 or 3 genes involved. So backcross trees are certainly more resistant than American. They're not as resistant as Chinese because it's way more genes involved. Well, there's an argument about how many more. I mean, it could be hundreds, it could be 6 or 7. We don't really know yet. We would have had to do a lot more breeding to get all those genes concentrated in fewer individuals. Yes. So you start with like American and Chinese, you make a hybrid. Okay. So a backcross technically is another hybrid. So it's a backcross hybrid, but you start with the target parent, I guess you'd call it, like the American in this case, put pollen from that American dad onto a hybrid mom. And then the offspring on average would be 3/4 American. And then you do another backcross with those progeny and you get 7/8. And so you slowly, making it, you're selecting along the way for resistance. So you're hoping to get a tree at the end of the process that's 15/16 or even more American genes from the dentata species, and but brought the resistance along with it. And they do look, that part, I mean, these backcross trees, in fact, I believe this is one of them, is actually like 99% American when we genotype it. So it's even better than 15/16. It grows very American, and it has intermediate blight resistance. Enough that it survives in this long, but it's probably not enough blight resistance to be ready for total restoration, the ultimate goal of restoration. So anyway, so the chapters do these breeding orchards. So we have the blues all over the place here in the state because we really wanted to get the diversity. This is one of the reasons why we had these chapter orchards, is that we could capture the full genetic diversity of the species in the breeding program. And then we also have these larger green areas, which are seed orchards, and they're concentrated for the Virginia chapter in Northern Virginia because we had good sites for them. And that's where we take best individuals out of these breeding orchards. And there's one breeding orchard right here, which is the Catawba orchard, not far from here. And we put those in these seed orchards. And that was to be the production orchard. And it can still be, but it doesn't have quite the resistance that we want. So we're continuing to work on that. Then we also have some restoration plantings around the state that are chapter projects. And one of those is actually near here as well. And I'll have a picture of it. It's the Johns Creek Mountain, which is up near Mountain Lake or close to Nida State Forest, if you know where that is, where we planted some of these backcross trees. So I'll show you some pictures of how the backcross trees do in the woods. But as a chapter, we've planted 36,000 chestnuts at least. We have about 21,000 surviving in these orchards, and they're flowering. Not all of them, but many of them are flowering, and they're open pollinating each other, or we're doing control pollination. So we're keeping the genetics going and advancing. We're kind of fostering evolution in that way. There's also Meadowview, which is the national down near Abingdon. It's a Meadowview Farms, which is where National concentrates its breeding work, or headquarters. I'm not sure if concentrate is the right word, but it's a great facility down there. They have an excellent greenhouse. They have lots of orchards, and it's the we're blessed to have it here in Virginia, but other state chapters have a similar system of these remote, widely distributed orchards. And so that's what a lot of what we do. So let's see. Next. A germplasm conservation orchard. So fancy name for wild type American owned. So we go collect these large surviving Americans somehow, and we plant them there, both to keep them alive, but also it's hard to pollinate these trees that are out in the woods. So it's a great place to have a variety of trees that you can just pollinate quickly or collect pollen from quickly. So yeah, we have those as well. So this is actually the Catawba orchard. So if you don't know where that is, McAfee's Knob parking lot, you go down the hill about a quarter of a mile and it's on the right, which is best you can describe it. Although you'll note you need to know where to turn, which is not easy coming down that hill. And Carl Apsher, I think many of you do. He was really the leading force in this area. He arranged with that's on the properties on the Virginia Tech Sustainability Center property, and it is a breeding orchard of the Nanking line, which was one of the selections of Chinese that were used to bring resistance over. And it had a number of problems in the first few years with voles and just poor growth, but Carl kept at it. It's got a deer fence around it, and we have a number of trees, particularly the first few years of trees that have been big enough to start evaluating for blight resistance. And in these breeding orchards, the protocol was to inoculate them with blight and measure how the cankers grow. So they all are get the same kind of challenge from the blight. And we've recently just evaluated those. We inoculated them a little over a year ago. And the next step actually in that orchard is to cull out the ones that kind of fail the test and keep the better ones and let them open pollinate each other. You can also see, and I'm not even at all the way up to the top of the orchard when I took this picture, but there's more room there that we can plant new trees in as well, either large surviving American progeny or other crosses that we might do. So it's a nice orchard. Carl put a lot of work into it, and I think we like to keep it going and be of use to the program. We also actually here he's at, I believe this picture on the right with Carl is at Johns Creek Mountain when I think it might have been when the first measurements were made. And I'm not totally sure of that. So what is our work ahead of us? So we are, so I've mentioned the back crossing program, was a partial success. And that was a form of traditional breeding, transferring resistance genes from Asian chestnuts, and also finding resistant genes in wild type American chestnuts. And the American Chestnut Cooperators Foundation has pursued that for a number of years. And Dr. Gary Griffin from Virginia Tech, who taught forest pathology to my students when I was teaching here, and he was involved and was saying from at least 1980, doing his early hypovirulence work, was there at Lassane, and then some of the American breeding work that he and his wife Lucille did was also various places, but included Lassane. And those things I think are still worth pursuing, the resistance from Asian chestnuts and finding resistant genes in wild type Americans. And one new technology that can help us with traditional breeding is called genomics or genomic selection. So we can genotype these trees now, do a whole genome sequencing of them, and use that in a computer model, a big mathematical model, to predict whether they should have resistance. I mean, we have to have both the genetic data and what we call the phenotypic data of blight resistance, how blight resistant they are. And we can come up with a model that predicts. And that seems to be a very useful tool. And that can help both the backcrossing program and the wild type American program. And then there's also, lots of bioengineering you've been hearing about, from gene insertion to CRISPR gene editing. And those techniques, are really making advances very quickly, and we're not quite up to speed, but we want to. And the one you may have heard about most was something that was started over a decade ago up at State University of New York School of Environmental Sciences and Forestry, was inserting the OXO gene, which is the blight creates, it kills, the blight itself can actually only eat dead tissue. So it gets in the bark and grows and produces oxalic acid and possibly other chemicals that end up killing or causing cell death. And then it can grow its hyphae into those dead cells. And so the obvious thought was, and that's a common fungal pathogen, and a lot of other plants have developed this oxalic acid or oxidase enzyme that detoxifies the oxalic acid. So that seemed like an obvious thing that would be great to have in chestnut. And so they inserted it with an agrobacterium insertion method. It's also what's called a constitutive promoter, which means that gene is always expressed. It's always being expressed. And the one they really liked was the Darling 58. And that actually got to the point, and it's still in the point of aphis deregulation, as they call it, being approved for further use without highly controlled situations. And also, one of the things we're looking at now too, because that gene actually came, where did it come from? Wheat, yes. And in fact, people were concerned about it doesn't have any, anyway, so another way we're looking at other genes that might be in what we call cisgenic species, which we're looking at other species in the Castanea genus, so American, Chinese, Japanese, and possibly others. And so we can use, so that's bioengineering using genes from other Castanea species. So we were very hopeful with Darling 58. Let's see. All right, so the progress. So we finished the third back crosses. It exhibits the third backcross generation exhibits a wide range of resistance from poor to pretty good. We are working on we're using genomics to select the very best in that population to come up with additional crosses, what we're calling best by best, very simple term, but where we can estimate, we can up those percentages or those ratings of blight resistance in a population. And then we can diversify it again later. So the transgenic approach, so I mentioned the SUNY ESF effort. When we started evaluating these trees, and one of the challenges of dealing with bioengineering or actually breeding in general with trees is they are long-lived trees. So we're looking for blight resistance that's going to be durable for the whole life of the tree. And there's no way really, other than growing them for decades, to know that for sure. And we also are looking for, as a restoration species, we want the full genetic diversity of the species to be restored. So we can't just deal with clones. We can't just narrowly select for the very best trees and plant them everywhere. We also have to worry about them being competitive in the forest. It's not a managed crop where if it grows a little slower, not necessarily a big problem. If it grows slower in the forest, it's never going to make it into the canopy and flower. So there's a lot of, so those are actually what we were finding with Darling 58 was the resistance ranged from very good to none, which we not sure why that was the case. They would test positive for having the gene, but the gene was possibly silenced because it was always being promoted. That's one problem with using a promoter that is always expressing the gene. The plant's like, why am I wasting all this energy on producing this oxalic oxidase, and then turns it off? Or it's treating it, another way of thinking of it is kind of treating it like a virus. That could have been happening. We don't know that yet. We also were noticing growth penalties of having the gene. And we were investigating some of that and we were also trying to further along the breeding at the time. And we were finding out that we were having trouble crossing D58 containing trees with D58 gene in it with another a tree with D58 gene in. We weren't getting homozygous offspring, like the 1/4 you would expect. And could be a number of reasons why. Could be the oxalic acid is expressed in the nut itself. So that could be, so even in the embryo, that could be a problem. And then when we were investigating that, or one of our cooperators investigating that, they found out they were looking at the gene and they couldn't find it in the place it was supposed to be. And it turned out that there was a lab mix-up and D58 was actually switched with D54 in the lab. And D54 was an earlier event that was rejected because it overwrote an existing gene, which is involved in drought tolerance. And so that possibly explains the growth penalty and other symptoms we were saying. So I'm really unfortunate that because this actually happened, this mistake in the lab actually, I think, happened back in 2016, and we didn't know about it till now. So we've wasted a lot of time on testing and trying to get D54 through what turns out to be D54 through the process. But in that, we decided, at least the American Chestnut Foundation decided that, well, certainly D54 has too many problems to release it as a restoration tree. It might still be worth further study. We also think that we're better off looking at non-constitutive, what are called wound-inducible promoters that only turn the gene when there's a wound. And we might also look at other approaches as well. So we're continuing the transgenic efforts, and we just don't think D54 is ready for prime time and at this moment. And we're working, we're expanding our efforts, or cooperate. I mean, actually, we're reaching out to people who have been working with us and encourage them to do more. A good instance is University of Georgia, which is a really good lab for somatic embryogenesis, which is you take little embryos and multiply them, and allows you to do a lot of gene insertions over a lot of them and pick the best ones. So that's like the first and really the hardest part of the transgenic process. And they're having really good results. And I'm proud that some of those trees, actually one of those trees you saw in that picture of Hossein is, we did 2 American by American trees in that planting there, and they are some of the sources of the best somatic embryogenesis lab that University of Georgia has right now. So yeah, when you do this work, you get to know these individuals. Oh yeah, 762. She pulled through down there at UGA. So another lesson I think we learned from this is that multiple genes are involved, probably, that resistance is a complex biological process, and it's going to be probably many cycles of breeding. We can, we may have, even if the backcross trees that are ready for partial restoration, not fully meeting the requirements of restoration trees, but we might start getting those out in the landscape and learning about how they proceed. We can start further the traditional breeding, further the transgenic work, and start building successive generations of trees that are more and more resistant, more and more competitive in the environment. And I'm taking way too long. Let's see. So, I mentioned trees are long-lived, so we need to have greenhouse and orchard tests. We need to test the effectiveness, the resistance, and potential side effects, to test expression and heritability of the gene too, because we want these to reproduce on their own. So you could have a transgenic tree that you could create and clone and perform well, but it may not be able to reproduce on its own and get out into the forest. Forest testing to look at resistance and fitness in the different genotypes and environments. There may be different gene combinations that do better in New England than they do in Georgia. They may do better at high elevation and low elevation, drier sites, wetter sites, stuff like that. Forest testing to validate the gene and the trait are safe to the tree and the environment, and the trait is effective and durable. So it lasts many years. So one thing we've learned as well is that the existing regulation system is so the APHIS was the first hurdle that you have to go through with a plant, and they really only look that it poses no plant health risk. It doesn't look at effectiveness. When I asked somebody about that, they say, well, the market will take care of that. As long as it's safe, you can try it, and if it doesn't work, that's on you. So I think that was a lot of confusion. People were thinking, oh, well, if APHIS approves it, it's ready to go as a restoration tree. We realized that we didn't have in place a good system for testing effectiveness. We were trying, but not as extensive as I think we should have done, and as quickly as we should have done. EPA approvals next, and they just say there's no public health or environmental risk. And so that process is the next step, which D54 never got to, or hasn't got to yet, I should say. And then since it's a food, FDA has to prove it that it's a safe food. But no agency judges whether the trait is effective in conferring resistance, has genetic fitness, nor the trait is heritable. That's wrong. It should be heritable and durable. And so we're working on a setting up protocol with other scientists that would set the standards and testing you have to meet to get a restoration tree kind of approved by at least the American Chestnut Foundation. So I'll go past this. So here's the John Creek Mountain Test. So here are trees that Carl, and I think you were here to help some of it as well, Brenda. This is, I did get one of the best trees, but on the left is David, who's 6'3", measuring a 30-foot tree that's 12 years old and about 7 inches in diameter. And it has, I can see a little blight canker on it. There's definitely the trees in this planting had about 60% survival at age 12, which is good, certainly for chestnut. It's actually pretty good for any kind of hardwood forest planting. And this is one of our best tests. And Carl deserves a lot of credit for that as well. And these trees were flowering. Some of these trees, like that tree, were flowering, producing burrs. And the bears found out. They haven't forgot about chestnuts. So they were climbing the trees and breaking the limbs out of the top of the trees to get at the chestnuts. So I guess that, I mean, in a way, that's a good test of our restoration. So yeah, getting them out in the forest, I mean, they're going to face it just like oaks and everything else does. They have deer browse to deal, bears to deal with, competing vegetation, the native tree sprouts, they're competitive, hopefully against them and tulip trees, but they were very fire adapted. And the forest has been, I don't know if you've heard the term mesofied, they're more moist and humid than they used to be before, when there was more fire and more open woodlands and not as many invasive species. Certainly in a lot of places, autumn olive and oriental bildersweet, we're fighting those all the time at that Lasagne State Forest. Those trees would not be surviving if we hadn't fought those species over the years. And then there's barrier to natural regeneration, seed predation, shade levels, fire suppression. We're going to try a prescribed burn at Lasagne State Forest sometime this spring, as soon as conditions are right. And that half of that old hybrid orchard to see, because one thing we've noticed, there's no natural regeneration in that orchard. It's got a thick understory of poplar and maples and fair number of autumn olive and everything like that. And it's just not conducive to, there's plenty of burrs and nuts on the ground all the time, but very little. I mean, we found a few seedlings here and there. Pop on that. So one thing everybody always asks, so I'll get ahead of you. So where do I get trees? Well, a lot of the trees from, the Lasagne Forest, the Lasagne orchards belongs to the Virginia Department of Forestry. We cooperate with them very closely, and we help them harvest. We harvest the control crosses and some open pollinated cross of nuts that we want for research purposes. But we generally, several thousand nuts are harvested there every year, and the rest go into the seed beds at the Augusta Forestry Nursery. And for the last 3 years, we've been selling those to the general public, both as mixed lots of either all American wild type seedlings from the American orchard we have there, or we call them hybrid backcross. They could be, it's just a mixed lot. It could be backcross 1, 2, or 3. It could be a really nice tree. You could end up with something that looks very Chinese. But they are, and if you're not familiar with that seedling sale, they open it up in October, and you can pick the seedlings up April 1st, or get them shipped to you at April 1st. You can still get on there and order a lot of species, but the chestnuts sell out within a few weeks. So look for October 1. If you're a member or want to get on our mailing list, we always send out a notice about that sale coming up. The American Chestnut Foundation sells wild type trees in March, coming up soon. I forgot the exact date it opens up, but if you go onto the ACF website, you can find that. And if you join at a C level membership, you get a certain number of chestnuts every year of the latest back crosses. I recommend getting Virginia sources, or at least other southern sources, because I didn't even mention the Phytophthora root rot, but that's a problem in the coastal plain and the Piedmont and a few places in the mountains, including at Lasagne State Forest. And we're working on developing that. And that seems to be a more tractable problem of getting genes from Chinese. There's fewer genes involved to give you resistance to the Phytophthora root rot as well. But whenever you're buying chestnuts, unless you're buying Chinese or something that's hybrid 50% Chinese, they're gonna get blight, and you're lucky they'll survive, but a lot of them won't. And if you're a place that's got wet, compacted soils, you might get root rot, and you're certainly going to have deer come after them. Deer love them; they're preferred browse. But But sometimes it works out, so, be hopeful for some success. Let's see. How can you help? Oops. Certainly, you're welcome to become a member. I've got a few magazines here that you can always just go on tacf.org and join as a member there. You can certainly support our efforts financially. You can volunteer, and it's kind of this group probably a great audience for volunteers because we generally need people. I mean, sometimes when we have planting, we can just get, kids, anybody who can dig a hole and put a tree in it. But a lot of our work is like control pollination, measuring trees, collecting data. And so you want folks who can handle that kind of work, that kind of concentration, and have an interest in it and enjoy it. Probably harder to find. So, we're measuring that Johns Creek test, and it's climbing up and down the steep mountainside and measuring trees. It's not, it's not for the faint of heart, but it's interesting if you're. But if you're just out hiking and you see a chestnut, there's an app you can download on Android or iOS, Apple or Android, called Tree Snap. And if you find a tree out in the woods, take a picture of it. It gets a GPS. It goes onto an online database. You can, even if you don't have cell service, you can upload it later. Some people like myself have access to your data. Nobody else has access to your data. But I can, I just this last weekend, I downloaded all the observations from Tree Snap, several thousand of them that were done, updated to current, and I make a map of it, and I actually have it on my phone, so I can go and look at trees that people report if they look interesting. You can plant trees, establish, we're looking often to establish new orchards. We're kind of in a holding pattern, like how we're going to do the next orchard. So not one, not this spring, but, possibly, down the line. That can be a few acres to, 20 acres or something like that. If it's the right kind of place, the right kind of, we have a need. I encourage you to look at tacf.org and particularly the VA page, but there's a lot of other good information there about all this science that I've been talking about. Pretty much all of it's there from what I drew much of this from. And then email us at vachestnut@gmail. So that's a little over an hour. Do you want to take questions from the audience or on Zoom as well?
You're talking about the trees that are associated with the chestnut labor book. I find it odd that white oak wasn't mentioned in that.
Yeah. I mean, actually, Lucy Braun talks a lot about white oak and where it was. I mean, it was more prevalent in the valleys, and I mean, it was certainly found some with chestnut, but yeah, she actually has more of like a white oak type that is. Now, chestnut oak is found. That's like, that was, I don't know why many reasons why chestnut oak may be named chestnut oak, but the leaves look similar, but it also grew as they grew together all the time, same kind of sites.
So planet chestnut, kind of some seedling from home, but now it sounds like you try to put them on a higher, drier place, not in a wet, lower.
Yeah, that's generally true. I mean, chestnut does well on well-drained soils. I mean, I've seen some like stream terraces that are well-drained that may even occasionally flood. They may grow well, but generally you don't want them sitting in water for long periods of time. In fact, even in the greenhouse, there's a more danger of overwatering them than underwatering. So. And then particularly if the soil has Phytophthora cinnamomi in it, you may not find out it has Phytophthora problem until you have a really wet year or something. So, yes, sir. How long to get across? Okay. So,
How long do you have to wait to do the back crop?
Well, you have to wait for the progeny to flower. So when you plant them out in the outdoors, when you plant them out in a natural environment, you can get flowers on vigorously growing trees 3 and 4 at ages 3 and 4, but reliably in an orchard, you're looking for like ages 7 to 10. So we kind of said, you know, an orchard generation is 10. Now we do put them in greenhouses, highlight chambers, highlight growth chambers, and you can force them to produce pollen in a year. The female flowers, not so much the first year, but some in the second year. So there are ways of accelerating that breeding, but yeah.
Is that so somewhere somebody had said we roughly would have having 15, 16 to 31, 30 seconds. That doesn't sound like that could be right.
We have them, yeah. We have.
That would be 5 or 6 back.
No. The first back cross starts off with a 50-50 parent and 100% parent. So the first back cross is actually 3/4. So the second is 7/8. The third is 15/16. And we have fourth back cross trees at Meadowview and other places. And so, so yeah, that and those trees really are, you know, you can't tell from the leaves without looking at a hand lens, find a few stray hairs that are carried over from the Asian species or something like that. So. I don't know, speak up if you're on, okay. So I actually have a couple questions in the chat. Has any effort been made to kill the blight? Oh, yes. So, I mean, some fungicides will work, but it's not practical for restoration to go spraying fungicide all over the place. There is a disease of the blight itself, a viral or an RNA virus, which infects the blight and makes it hypovirulent, weaker. So trees can more likely survive it. And that's actually used fairly effectively in Europe to keep orchards going and even some forest plantations. It's actually Gary Griffin, Dr. Griffin from Virginia Tech was really did some of the really earliest and most important work on that. We found the problem in North America is that disease can only really be spread from blight culture to another blight culture if they're compatible. And there are so many varieties of the blight in the United States because so many different Japanese, Chinese trees were brought here. And for some reason, there's fewer, they're more manageable to do that. And it's still a lot of work in Europe. Those are probably the two main things. The other ones, how did Native Americans manage chestnuts? So did they use fire, for example? Yes, there's good records of them. First of all, fostering chestnuts and probably fostering chestnut growth, the wild chestnut grows, but also probably planting them. But there's good accounts of them using fire to help harvest because we harvest generally we get in bucket trucks and grab burrs off the trees before the nuts drop for a couple of reasons. I mean, one, if it's a control cross, we don't want them to get lost. But it also, they're less likely to have worms in them. And they rot pretty quickly on the ground, actually. So, but apparently and commercial orchards keep the ground very, very clean and come and collect them right away as soon as they drop. But the Native Americans would use fire as the chestnuts were dropping, and a light ground fire, and it would kill the weevils and prevent them from coming back the next year. And it wouldn't destroy the nuts, but lightly roast them because the burr would protect them. And so they also certainly use fire to manage forests to promote nut production in general, not just chestnuts, oaks and hickories and beech. Yes, sir.
Is there any thought, not by you, because you're going different route, by others using D58, the actual D58, and proceeding with that and making another stab at that.
Yeah, I think SUNY ESF has gone back. Now, the last I'd heard, they had found just a handful of plantlets, not trees out in the woods, those trees in the lab, that they were gonna have to start over with, which, you know, it's a long process. So,
Justice ecology, I found this on the web and it talks about the research at SUNY Syracuse.
this is, this, from the title, this looks like it's about this whole issue. Yeah, I haven't read this particular, but, okay, thank you. Yeah, I mean, it's certainly unfortunate in many ways that this first transgenic effort ran into these roadblocks. But one thing that the APHIS process did very well was go through all the plant health risks. And, you know, the inserting this one gene, which is a common gene in a lot of plants to deal with oxalic acid, you know, showed no negative plant health effects. So in terms of, yeah, I guess in terms of plant health risk, so it showed the growth thing, but that could be for a number of reasons, the gene deletion or whatever. Yeah, so, you know, it hasn't soured us against transgenic efforts, and, you know, none of the hazards that people think or may happen with transgenic trees did not occur with this. It certainly didn't make it more aggressive in a weedy species, unfortunately. I mean, Chestnut was kind of weedy to begin with, and we're hoping to get it at least back to close to being that aggressive grower tree. So, and, you know, there was no, I mean, oxalic acid, I mean, the oxo gene, like bananas, strawberries, you know, wheat, it's a good chance you ate some of it today. So, you know, we feel it's a safe way to go with, you know, proper precautions. So,
There's
I
one
don't-
more. There's
Yeah.
one more question in the chat. It asks, did Gary Griffin have a chestnut research place on Craig Creek Road about 25 years ago?
I'm not exactly familiar with that one, but I would think so. They're all over the place, and that'd be yeah. I actually think I have heard of that one, but I'm not that familiar with it. So you could contact the American Chestnut Cooperators Foundation. They would know. Oh, ooh, I forgot my last slide. So anybody who wants to come up next week to Cremora, Virginia, near Waynesboro, can help us lift and grade some of those seedlings you saw in the nursery. And then on March 23rd, we're having a tour of Lasagne State Forest, and I'll be sending out more information about that. And then we're going to be back. Some of us will be back here in May or June to take a tour of the Catawba orchard and maybe do a little cleanup work there and maybe look at some of the results. And then in June, we'll be out pollinating, and we don't always know what our priorities are, but people are welcome to come and learn how to do pollination. And then, you know, if there are isolated trees around here that, you know, would be useful to pollinate, we can have people do that. It's not very hard. You find the female flower, you emasculate the flower by taking all the male catkins off, and you put a bag over the twig, and then you come back later with some pollen and just wipe it on the styles on the flower, and put the bag back on and come back in September. So.
You were a betting man. Where would you place your highest bet on what area of this? You got a bet?
Yeah. So, yeah, my bet is not going to be one thing. Yeah, we use the cliche, you know, the OXO gene D58 was kind of a silver. We viewed it as a silver. A lot of people, including, you know, at points, you know, a lot of us did, viewed it as a silver bullet. This was going to solve the whole problem. And, and looking back, we're really kind of naive that, because plant pest host relationship is more complicated than that. You just can't just detoxify one thing and solve it. It's my feeling. I mean, maybe I'll be proved wrong. But, I think it's got to be a mix of geneticists talk about qualitative or major genes, like just like one or two genes that control something really obvious in the phenotype. And we may need some of those major genes, something like OXO. But there's also what are called quantitative genes. There's a whole lot of genes that work in concert to deal with different aspects of these, in this case, disease resistance. And we probably need a mixture of those to work well. Because even Chinese will, if it's under enough stress, it'll succumb to the blight. So, they don't have a perfect system. Chinese doesn't have a perfect system figured out. They're not totally immune. They're very strongly resistant. But, it's going to be, I think, a continuous improvement cycle that, we're going to have trees. You could argue now that the backcross trees are good enough to start with restoration. Some would disagree with that. I think they are, at least to start learning more about restoration process. Even if one thing that Chestnut is actually very good at doing is thriving under coppice management, which is where you grow the trees for a certain time. Not necessarily, well, there's a short coppice where you don't grow them, they're just firewood or something, or pulpwood, and you cut them down and they resprout and they grow faster the second time. And you can just keep harvesting them like that. And it's not the kind of chestnut forest we're at the end, but we could probably do that with I think we could do that with the backcross trees. And then as we get better and better resistance built into the genome, we can, extend it to different kinds of restoration. So, yeah, I bet it's going to be a mix. Wow. Thanks for all the time. Any other questions, Warren, or I see anybody else in chat? I do not.
Nothing else in the chat. I thought that was well said about what could be a restoration tree, and especially what genes we would maybe have to target for like another GMO tree.
Yes, thank you. If you don't know Lauren, you all know Lauren, I assume. Yes, and she's works for ACF and our lab manager down at Meadowview. So, anything else you wanted to add, Warren?
I guess I'll point out that as far as the OXO tree, a lot of people were very disappointed that it didn't turn out to be the silver bullet we thought it would be. But I think that we are kind of on track with how crops have fared with genetic modifications from genes from other trees. And so we're kind of maybe a little bit behind because we're still in tree time, and they are working with annuals or plants that have a much shorter lifespan.
Yep.
But we're kind of in a transition period where we get to look more towards the future with like newer technology. And that's what I'm really excited about.
Good. Yes, well said. Yeah, and I think just newer technologies. Yes, and that's Lauren appreciates that, but I definitely appreciate it because I took genetics over 30 some years ago, and what's available to us now is just unimaginable. It was unimaginable back then that we would be able to know this this much about what's going on in the genome and make judicious changes in it. So I agree. Thanks. Yes.
You said something about an internship.
That's yes. Yes. So yes, we in honor of Carl, we a number of people donated, and the chapter matched enough to fund it at least for 2 years, and we're going to continue to raise money to keep that going over the time. And Cassie Stark, who's the regional science coordinator for the American Chestnut Foundation, she's stationed in Charlottesville, and she is reviewing applications, and I haven't checked in for a week. They're actually closing tomorrow for applications, but she had over a dozen or more, I think, last week, and she hopefully has a few more. And she said there were a lot of good ones. So we hope to have somebody selected in the next few weeks and get them started in as early as May. It's a summer internship. Yeah, looking for a student or even a grad student. Yeah, and so we want to get them started in May so they can be involved in some of the restoration plantings we may still be doing in May. Certainly the pollination season, because that's kind of our busiest time, and you need people who not know what they're doing and can get up tall in a tree and and do that. And so we need somebody full time who can, because in June it's like you're depending on the phenology of the tree, when to get the bags on, and it's always go out and say, oh, they're not quite ready to bag, and then come back 2 days later, oh, we almost missed it, and then come back, put the pollen on and all that. It's you have to pretty much block out every day in June to be able to get the work done. So so yeah, we're really hopeful to do that. I mean, there's an intern, Lauren, there's an internship position at Meadowview as well, correct?
Sorry, I'm still muted. Yes, there is. And I don't know if we've started hiring for them yet, but it will probably be posted on Instagram when the position opens, just like Cassie's position was.
Yes, I think, yeah. Okay, great. So you can send it, you can go on tacf.org and donate to a chapter, or maybe just in the note, or even it actually all ends up going to the national coffers, but if in honor of Carl Absher might do it as well. So just that's the way a lot of them did it last time, either way. But yeah, that would be very helpful. And we'll get that appeal out every year as well to keep and report on what the intern did each year. And certainly this intern we certainly hope in May and June that they'll be there as well and work. That's one of the things they'll do is we have pollination work here, send that intern down to supervise that. So, well, thank you.
Thank you.
Thanks, John.
All right, Lauren, I can turn it back, or you can take it back. What do I need to do? Stop share?
You stop
Okay,
sharing.
I
I
did.
will
Okay.
unpin you. So it's just general now.
Okay,
Thank you.
you're welcome.
And then you can put Bob back on to give closing remarks.
Okay.
I was fortunate enough to go to the movie they had a couple of weeks ago that was called Clear Day Thunder, I believe.
Yes.
It was an excellent film. So this was great to have this follow-up talk coming back from that presentation. So if you have a chance and you see a movie with this unusual name, Clear Day Thunder, check it out. And about halfway through, they'll actually tell you why the movie's called Clear Day Thunder. So no spoiler alerts tonight. I'm glad we had our speaker tonight. It was excellent. I believe next month, our speaker will be talking about ferns. Is that correct? I'm getting nods of yes, so I did remember correctly. And yeah, and so yes, a couple of things I just wanted to mention really quick is check your emails. Jennifer puts out a pretty good list of immediate events that are coming up. You'll also see that the newsletter listed events going quite a bit out all the way to October, which so you can then, as you find about these, you get the definite details coming up from Jennifer's email later on. And we have a bare root plant sale happening, which Beth Umberger is running, which is if you're interested in native plants, it's unlikely, not no, but unlikely that we're gonna have the native plant sale this summer at Seeds. Because we need a chair, someone to run the project. So no one stepped forward yet, but if they do, we could still have that sale. Otherwise, check with Beth about the bare root sale that she has organized. And I think that's just about all of my notes for this evening. So I'm glad we're all able to make it. There's quite a large group tonight, and should be another excellent presentation next month. Thank you.
Got to mention this tree.
Yeah, we